Cold Spring Harbor Laboratory is a National Cancer Institute designated cancer center, and we have been so for the past 25 years. Over 50% of the research at Cold Spring Harbor Laboratory is focused on cancer research. And in addition to breast cancer, we're working on a large number of other cancers, including brain cancer, pancreatic cancer, leukemia, lymphoma, prostate cancer, melanoma, ovarian cancer, and lung cancer, to name just a few. Our research is consistently rated highly by independent surveyors, and in fact, we are rated #1 by Thomson Reuters for our efforts in molecular biology and genetics, and that's due to the impact that our research papers have in their respective fields. So not only are we doing fantastic research right here in your local neighborhood, but we're having a tremendous impact throughout the world in cancer research as well as in research in other areas. I think it's very appropriate tonight that we have this discussion on breast cancer, as all of us have a family member, friend, neighbor, or colleague that has been affected with this disease. And while we have made terrific strides over the past 10 or 20 years at trying to impact breast cancer, we still have a long way to go. And so the way we've structured our discussion tonight is to have 3 short talks. We'll first hear from Dr. Lara Weiselberg, who is a clinical professor at North Shore LIJ, and she'll talk to us about the current state of the art in treatment, diagnosis of breast cancer, and the mammogram controversy. That will then be followed by 2 research talks from investigators at Cold Spring Harbor Laboratory, and they'll tell you approaches that they're using to make the next big breakthrough in trying to move our understanding and our treatment of breast cancer ahead. One of the talks will be Mikala Egeblad, and she will be showing you the way she's addressing breast cancer using imaging-based approaches. And the third talk will be by Tonks, and he'll be showing you how he's using biochemistry to try to address having an impact in breast cancer, and how his approaches are really revolutionizing the way we might be able to treat breast cancer, and taking approaches that Big Pharma has seen not fit to fund. And so this will be a very nice demonstration of how basic research at an institution like ours can really make a big difference in trying to help individuals that are afflicted with this disease. So without further ado, I'd like to introduce our first speaker, and that's Dr. Lara Weiselberg. She's a clinical associate professor at Hofstra, North Shore LIJ School of Medicine. She's also at the Monter Cancer Center of North Shore LIJ, and her talk tonight is going to be Breast Cancer, Past, Present, and Future, a Clinician's Perspective.
Thank you very much. Can everybody hear me? Thank you very much for the opportunity to speak at this beautiful institution, and I'm sure that we're going to be seeing some very elegant research presented after my presentation. But I'd like to give you my perspective with 35 years of clinical experience practicing oncology, and mostly breast cancer oncology, how all of this impacts on our day-to-day practice, and I think everything is connected, as they say. So let me just, can I close this laptop? Thank you. All right. So breast cancer has been around for a very long time, since there's been recorded history. This particular painting hangs in the Louvre. It's Bathsheba with King David's letter, and it was painted by Rembrandt. And shortly after this painting was completed, the model who sat for it passed away. And when we look at it, actually, you can see that this breast is a little indented. Perhaps the area under the armpit is a little full. There's some swelling here above the collarbone, and actually this model was suffering from the advanced stages of breast cancer. So wherever you look, you can find some breast cancer. Breast cancer, of course, is a very important disease. It afflicts a lot of women. It's the #1 cancer in incidence in women in the United States. So the American Cancer Society estimates that in 2014, there will be 232,000 cases, new cases of invasive breast cancer in women, and approximately 2,300 cases in men. And we shouldn't forget that breast cancer also affects men. There will also be 62,000 cases of in situ carcinoma, and 40,000 women will perish from breast cancer, and about 400 men will pass away from breast cancer this year. Breast cancer is only surpassed by lung cancer in the cancer mortality of women. It's a very, very significant disease. So while we're getting better, we still need a lot of room for improvement. So how far have we come? Well, there are a lot of aspects in which our knowledge is rapidly increasing. We better understand the biology of the disease, and I think we'll see some of that today. We have advances in treatment. We are able to personalize treatments better for each individual woman. It's no longer one size fits all. We are beginning, at the very beginning, strategizing to prevent breast cancer. And everything we do, we owe to research that's been done from the bench in the lab to the clinic. So in the beginning, this gentleman is William Halstead. He was the first professor, chairman of surgery at Johns Hopkins University at the turn of the 20th century. And he had a theory about breast cancer, that breast cancer started in the breast, and then it spread to the lymph nodes under the arm, and from the lymph nodes under the arm, it spread throughout the body. And if you could remove that breast cancer effectively, you could cure breast cancer. So he developed an operation, which is called the radical mastectomy. And nowadays, it's a pretty brutal operation. You don't see that anymore. The breast, the muscles, and all the lymph nodes under the arm are removed. And it causes tremendous cosmetic deformity, and it leads to lymphedema or swelling of the arm very frequently. But his theory of breast cancer is called the Halstedian theory. It spreads in an orderly sequence. And this was pretty much how everybody thought about breast cancer till another surgeon came along in the 1970s and 80s by the name of Dr. Bernard Fisher. And he had another theory about breast cancer. And his theory was, in some women, breast cancer was a systemic disease from the very outset. So at the time of diagnosis, there were already microscopic cells hiding somewhere else in the body. So doing a bigger operation isn't going to cure those women. In fact, what you need is to treat the rest of the patient, to give them a systemic treatment to kill those microscopic cells. And based on his research, the lumpectomy was developed, and the era of breast conservation surgery came about. He was also the first person to use chemotherapy after breast cancer and demonstrated that it reduced the risk of a reoccurrence. So even though he was a surgeon, no offense intended, he was an innovative thinker and thought really beyond the knife. So what's going on in terms of improving the treatment of breast cancer locally? Well, now we have breast cancer conservation surgery, which is appropriate for the majority of women with breast cancer. There are still some women who are better served by mastectomy, but the vast majority will have the same outcome with a lumpectomy or a mastectomy. For those who have a mastectomy, breast reconstruction is unbelievable. If you see some of these reconstructions, it's hard to believe that they're not real breasts, sometimes better than the original. And then sentinel lymph node biopsies, just taking out the first lymph node that drains the breast instead of taking them out all of them. And in many women, this is all that's necessary, and it has spared millions of women from suffering from lymphedema. And then, of course, there are many developments in radiation therapy to the breast, which we usually use after a lumpectomy. There are many aspects that are currently under study. We're looking at drugs to make radiation work better. We're trying to speed up radiation so that we don't have to spend 6 weeks administering it. And we're looking at groups of patients that may not even need radiation after breast conservation surgery. So we're not resting on our laurels, but we're constantly looking beyond, trying to improve it. And everything has to be done by well-controlled research, because you don't want to do something on an intuitive basis. Well, breast cancer, we used to think of breast cancer as just breast cancer. It's one disease, but it's not. It's a family of diseases. And we don't know exactly how many members there are of this family, but there's at least 4 significant types of breast cancer. There's the HER2/neu positive breast cancer, which I will explain in a minute. There's something called basal-like breast cancer, or triple negative, sorry, typo. There's luminal A breast cancer and luminal B breast cancer, both of which have the potential to respond to hormonal treatments. So in the olden days, when a pathologist looked at a slide, he would see the cancer cells, and then he would describe them. He would say they look very aggressive, they're growing into the blood vessels, they stain for hormone receptors or they don't. And that's how we would classify breast cancer. So we'd say it was poorly differentiated infiltrating ductal carcinoma. And they'd count the lymph nodes that were involved, and then the clinician would try to make a judgment about the best treatment. But we know breast cancer in one patient doesn't behave the same way as breast cancer in another patient. So how do we distinguish who's going to behave well and who's going to have an aggressive course? Well, it turns out if you look at the genes that are expressed in the cancer, they fall out into these 4 groups. So those cancers that are HER2/neu positive and basal-like are very aggressive, and the luminal A's and B's are less aggressive, with the luminal A's having the most favorable prognosis. So we shouldn't treat somebody with a luminal A breast cancer the same way we treat somebody with a triple negative breast cancer. We have to be more specific in our treatments, and we have to find treatments that work better for each kind of breast cancer. So this brings up the idea of targeted therapies. And targeted therapies are designed to interact with something within the cancer that's used by the cancer cells to grow. And hopefully, if we pick a targeted therapy, there will be less toxicity to the normal tissue. So this is really where we're going with our treatment. Well, the very first targeted therapy isn't very new. In 1896, George Beatson, who was a surgeon in England, did the first targeted therapy for breast cancer. And what he did was he removed the ovaries on a patient who had metastatic breast cancer. And fate being what it is, fortunately for us, the woman was premenopausal, so her ovaries were producing estrogen, and she must have had a hormonally responsive cancer because she went into remission. And the subsequent patients that he treated also had a favorable response. So we began to understand that targeting hormones could treat breast cancer. And this was really the very first targeted therapy. The next step forward in the use of endocrine therapy for breast cancer was developed by this gentleman right over here in the third gas mask from the left. His name is Elwood Jensen. And during World War II, he was a young chemistry student graduate. He enlisted in the Army, and they put him in the chemical warfare unit. And he so enjoyed this, he decided to pursue a career in chemistry, and eventually got his degree and spent his career at the University of Chicago. And while there, he discovered something, first in normal tissue, and then in breast cancer cells, called the estrogen receptor. And the estrogen receptor is that thing in the breast cancer cells that can bind to estrogen and be stimulated by estrogen and affect the breast cancer cell. So the final great moment in the endocrine therapy of breast cancer was the development of a drug that attacks the estrogen receptor. And that first drug was tamoxifen, which was approved by the FDA in 1978. And this is a slide that shows that if you give tamoxifen to an appropriate patient, after a diagnosis of early stage breast cancer, you will reduce the risk of a recurrence significantly. These are people on tamoxifen, lower recurrence rate, higher recurrence rate if they did not take tamoxifen, and their survival rate was also much better if they took tamoxifen. Well, things aren't that simple, and I'll leave this to one of the other speakers to discuss this. But it turns out everything interacts, so that the estrogen receptor can be stimulated by many other biochemical pathways within the cell. And this is a very active area of research in breast cancer treatment today. So Dr. Jensen, apparently I never met the man, but he was a very colorful guy and liked to end his talks with a limerick. And this one was, a lady with growth neoplastic thought surgical ablation too drastic. She preferred that her ill could be cured with a pill, which today is no longer fantastic. The other commonly used targeted therapy for breast cancer is HER2/neu. And there are receptors on the surface of normal cells and also cancer cells that can be stimulated by growth factors. And in 20% of patients with breast cancer, there are extra copies of this particular receptor called HER2/neu. And we know that HER2/neu, if it's overexpressed or amplified in a breast cancer, that cancer has a potential to behave very aggressively. And that was really an ominous diagnosis until about 1998, when a drug was developed called trastuzumab. I'm sorry, this is a drug company slide, so it has the brand name. But trastuzumab was developed. It's an antibody that attaches to the HER2/neu receptor and blocks it from functioning. And it has been a total game changer in the treatment of HER2/neu positive breast cancer. So as an example, this was one of the early studies that looked at giving patients with HER2/neu positive breast cancer Herceptin or trastuzumab in conjunction with their chemotherapy. And those patients who received the trastuzumab had a much higher disease-free survival rate than those women who didn't receive it. In fact, the recurrence rate was reduced by 50%. And in my 35 years of practicing oncology, this has been the single most dramatic advance that I have seen in the treatment of breast cancer. So it gives you faith. One of the other things we're trying to do is figure out who needs chemo and who doesn't, because you don't want that one-size-fits-all kind of, where the activists say slash, burn, and poison approach. So now we know that the luminal A cancers are probably not going to behave in an aggressive way. You probably don't need to use chemotherapy. Those patients are going to be very responsive to hormonal treatment. And a genetic test has been devised that we can do on the tumor cells of patients who have early stage breast cancer. And by early stage, we mean if they're hormone receptor positive and if the lymph nodes are negative. And more now, we're looking at people who have 1 to 3 positive lymph nodes for consideration of foregoing chemo. So there's this genetic test which looks at 21 different genes in the cancer. And they can generate a recurrence score, seeing whether these genes are mutated or not. There's some mathematical formula. I call it Shazam because I don't really understand it, but it gives you a number between 0 and 100. And if you have a low recurrence score, you are going to do very well on hormonal therapy, and chemotherapy adds nothing. If you have a high recurrence score, then you're still a significant risk for a recurrence on hormonal therapy, and adding chemotherapy will reduce your risk of a recurrence by 20%. Then there's all these people in the middle, which we're not really sure about, but there is a study that has been completed, and we're looking at randomizing people in this intermediate group to receive chemo or not receive chemo in addition to their hormonal therapy. And that trial is called the TAILOR-X. Hopefully we'll have a result in the near future. And there are many different trials ongoing looking at innovative ways to tweak our treatments, such as using many other drugs to attack HER2/neu, blocking the growth of blood vessels that supply blood to the tumors, using metformin, which is a diabetes drug, to interfere with those signals inside the breast cancer cells, and so on and so forth. Now, what about breast cancer risk? Well, we know there are certain things that are risk factors for breast cancer. Having had a prior history of breast cancer is the strongest risk factor. Having a family history of breast cancer, and I'm not talking about necessarily people with familial cancer syndromes like BRCA. They are very important, but they only account for 5 to 10% of breast cancer patients. Being overweight, being clinically obese increases your risk of breast cancer, particularly in postmenopausal women. Reproductive history is a risk factor. Getting your first period at a young age, going through menopause at a late age, having your first child after the age of 30, or never giving birth, all increase your risk of breast cancer. Drinking alcohol more than 3 drinks a week appears to increase the risk of breast cancer. Smoking. Well, smoking's not good for anything. Environmental factors, which are poorly understood but must play a role. Having had prior breast biopsies, not that breast biopsies cause breast cancer, but women who need biopsies probably have a lot of proliferative breast disease, so that's a marker for people whose breasts are not quiescent. And there is a mathematical model which anybody can access to assess their risk of breast cancer. It's called the Gail model, and it was developed at the National Cancer Institute by Dr. Mitchell Gail. And anybody who would like to use the Gail model just can go to the website cancer.gov, which is the NCI website. Type in, search for Gail model, and we'll bring you right there to do it. And it's good for women who are over the age of 35 and who have not had breast cancer. So anybody who'd like to assess their risk, it's our tax dollars hard at work. There was a study called the Long Island Breast Cancer Study Project, which was started 20 years ago. I can't believe it's 20 years, and it's a sign of my age. And it tried to locate every woman who had a diagnosis of breast cancer who lived on Long Island for a 1-year period, for a 12-month period. And in order to catch all the women who lived on Long Island, they went out to this geographical area, to all the cancer centers, to find patients who might have migrated out for their treatment but were living on Long Island at the time of their diagnosis. They also got controls who didn't have breast cancer. And they went into the homes of these women, and they did questionnaires about their lifestyle, the risk factors, taking birth control pills, things they may have been exposed to in their lifetime. They took samples of the air, the dust, the water in the homes. They looked at the electromagnetic fields around the homes, and they tried to see if there was something that made these women different than women who developed breast cancer. So sadly, we didn't come up with an easy answer. The suspects that they looked at, including insecticides, PCBs, which are a hot area right now, did not appear to be more commonly associated with people who got breast cancer or not. And also electromagnetic fields didn't seem to be associated with it. There was some suggestion that perhaps women who took aspirin got a little bit less breast cancer than women who didn't, but not strong enough evidence that I would advocate everybody should start taking aspirin. Obesity is definitely in postmenopausal women. Having a body mass index greater than 30 increases your risk of breast cancer. That's for a 5'4" woman being 30 pounds overweight. So that is a risk factor. And this is the scary part. This is a map of the United States in 1994 showing the percentage of people who were clinically obese, had a BMI over 30, and well, it shows the body mass, the average body mass in different states. You can see that no state had more than 20% of people who were obese. And not that many states had 15 to 19%. This is 1994. By 2009, there was only one holdout, only Colorado. I guess everybody was busy skiing and exercising. But every other state had at least a 20% incidence of obesity. And there's a cluster of states here where more than 30% of the population was clinically obese. And this is 2010, which is the last year we have data. And there are no more blue states. There are only 13 states left where less than 25% of the people are clinically obese. So we have an epidemic on our hands. So how can we manage our risk? Well, there's nothing you can do to eliminate the risk of breast cancer. I went to one website, they said risk factors, being female. That about sums it up. Try to maintain a healthy body weight, avoid hormone replacement therapy, which has been shown to increase the risk of breast cancer. Have your appropriate monitoring, limit your alcohol intake, and don't smoke. Women who are at very high risk for breast cancer, who carry mutations or have a high risk based on the Gail model, get certain recommendations, either careful monitoring. If they're very, very high risk, for instance, if they have a BRCA mutation, they could consider risk reduction mastectomies. But even after mastectomy, you can get breast cancer because they can't remove every last breast cell with the surgery. Or sometimes using medications can reduce the risk of breast cancer, such as tamoxifen, the drug that was developed to treat breast cancer, raloxifene, which is known as Avista, an osteoporosis drug, And these two drugs, which are called aromatase inhibitors, which are used to treat more advanced breast cancer. So all these drugs have side effects. You have to be selective when you use them, but they are options for very high-risk women. I wanted to approach the 800-pound gorilla in the room here, which is a study that was published exactly 1 month ago today and has really created a tremendous amount of buzz. And that study was a study that was published from Canada that looked at 90,000 women over a 30-year period to see whether mammography saved lives as compared to just clinical examination of the breasts. And that result of that study was that the number of breast cancers that were diagnosed was the same, and the number of deaths from breast cancer was the same. So this made a tremendous splash in the media, and everybody is talking about it. You couldn't turn on the TV for a week without hearing about it. But like everything else in science and in life, you have to look at the information critically. And there are some issues with this trial. The quality of the mammograms was felt to be not very good. Prior to going into the study, all these patients were examined, so those who had something abnormal in their initial exam were more likely to get a mammogram and skew the data. Other studies, and there are many other studies in the past, have shown the opposite result. So why is that? They stated that that many women had treatment for breast cancer that would never be dangerous. Well, I don't know how you define it as dangerous. That's kind of a loosey-goosey thing there. They said that women were afraid of being overdiagnosed. Personally, I'd be more worried about being underdiagnosed, but I'm not a normal person. I'm a medical oncologist. Quality of life was not evaluated, so women who were diagnosed perhaps at a later stage because it was by clinical exam, perhaps they had the same cure rate but had to have chemotherapy. So what was their quality of life compared to somebody who had an early stage breast cancer diagnosed and didn't need chemotherapy? So we need more information. I'm not saying the study is wrong. It may turn out to be right, and we need to have an open mind, but I don't think that the final word is in. So a caution to everybody, examine what you read in the media critically, and also what you hear in the broadcast media, think about it. Okay, the good news. The death rate from breast cancer was fairly stable until about 1990. It's going down. We are making progress. And we have many survivors that we can look up to as icons. So I'm going to stop here, and I thank you very much.
Our next speaker is Dr. Mikala Egeblad. Mikala is an assistant professor here at Cold Spring Harbor Laboratory. She's running an amazing breast cancer research program using imaging-based approaches to understand how the immune system is impacting the tumor and how chemotherapeutics impact that interaction. I'm delighted to say that Mikala recently received a highly competitive Department of Defense Era of Hope Scholar Award from their breast cancer program, and this grant is provided to individuals who are the best and brightest in their fields with a high potential of innovation and making breakthrough findings. So we're really delighted to have Mikala as a colleague here at Cold Spring Harbor Laboratory, and it's my pleasure to introduce her to speak with you tonight.
Thank you very much. I think we've lost the pointer here. Okay, there we go. So what I'm going to talk about today may be different from the kind of breast cancer research that you've seen before. What you have here behind me is one of these images that we have acquired from inside a living mouse. So we're watching in real time what's happening in breast cancer. What you're seeing here in blue, that's the tumor. In green, we have the inflammatory cells that you're going to see a lot more about in this presentation. And in red, we have the drug we are trying to treat the tumor with. And you can already see that one of the problems we are having is that the drug may come in some areas, but in other areas of the tumors is not getting in. So, as I think is the case for everyone in the audience here today, breast cancer is a personal issue for me. This is me when I was much smaller. And my grandmother. And my grandmother is a very typical example of the issue we are facing. Like most women, at the time of diagnosis, there was no evidence, no clinical evidence of a metastatic disease. But as is the case for 2 out of 10, the cancer came back despite surgery and despite adjuvant treatment, in her case, anti-estrogen treatment. And when it comes back, it can be very late, and that's one of the things that, of course, is concerning everyone that's a survivor with breast cancer. That it's really very hard to ever feel safe when you've had breast cancer. When it comes back, it often comes back in many different tissues, such as in the lung, the liver, the lymph node, or the bone. So in other words, it has metastasized or spread to other organs. And what we're trying to do in my laboratory is to try to address these problems. How do we make sure that the tumor that comes back doesn't metastasize and that it's not resistant to treatment? How do we get it so that we can cure everything in these early cases? So normally when a breast cancer researcher tries to attack that problem, what is done typically is to take out the tumor and try to generate cancer cells that we can study in the petri dishes in the laboratory and try to test different drugs and see which drugs kill the cancer cells and which drugs do not. And there's been major breakthroughs through that approach. We can go even deeper and try to do an analysis of the genes that are different in the cancer cells and find ways, vulnerabilities in the cancer cells, way to attack them. And you heard in the previous talk about ways that have led to breakthroughs, and you're going to hear another very elegant study after my talk. But the first thing that happens when a breast tumor is cut out is that a little piece of it is being brought to a microscopic slide, and a pathologist will be looking at the piece of the tumor. And what you can see here is that a breast tumor is more than just the cancer cells. It's a tissue. So we have the cancer cells here in blue, and interspersed between them you see these immune cells, inflammatory cells colored in brown so the pathologist can recognize them. And you can see there's also blood vessels within these tumors. So there are many different components in the cancer, and they are clearly speaking to each other. But when we take out slides and look at it in a microscope, it's just a still image. It's dead tissue and a snapshot. And we don't really know what happened before. What was the result of this interaction? We don't know what happens afterwards. So to try to understand that, we are taking advantage of some very basic research that were generated originally by a marine biologist. And they had no idea that this was going to help cancer research when it did it. And I think that's why it's so important that we support basic research. So they discovered that there's a gene in a type of jellyfish that make the jellyfish fluoresce. And this gene was isolated, and another group of researchers got the idea that maybe it could be transferred into organisms and make them glow so that you could follow the cells that had this gene in real time in living organisms such as the mouse you're seeing here. And a third group of scientists then figured out that they could modify these genes and get different colors, green, blue, red, purple, yellow even, and have different cells glow in different types so that we could see how they were interacting. So we have taken this approach and generated mice that have all these different colors in different cell types, and now we're putting the mouse on the microscope. It's live, so we have it anesthetized, and rather than watching a dead tissue, we are watching on the computer screen the live tissue here, and you saw some examples out in the hallway. So what you're having here is a tumor. The tumor cells are colored in blue. You have the inflammatory immune cells here in green, and you can see that they're moving through the blood vessel that we have labeled red. So what can we use this technology to? I told you that I really wanted to understand why tumors are not responding better to the treatment we have. So the first thing we tried to do was to take such a mouse on the microscope and treat it with chemotherapeutic treatment and see what happened. And the first thing we noticed is shown here. We have the drug, chemotherapeutic drug is colored red, and we can follow how it gets out of the blood vessel. They're colored in green, and you can see how individual cancer cells are lighting up as the drug gets inside the cell. And what is striking is that area of the tumors where we get a lot of drug in, and then there's areas where there's very little drug. And so it's not surprising that areas that see very little drug are not going to respond very well to the drug. We need a certain concentration, certain dose of the drug to kill them. So that is clearly one of the issues we are facing. But another thing we also saw and observed much later, what you just saw were the first hour after we injected the drug, came much later. So what you're seeing here is about 30 hours after we treated the mouse with the chemotherapeutic drug, and we watched them for up to 40 hours. You have here a region of the tumor. We used a trick, so all the dead cells are now labeled in red, and you can see we were quite successful. In this region, most of the cells were killed. But there's also some cells here that were not killed. They're alive, they're still glowing in the blue color. So in mice as well as in women, we need multiple doses of chemotherapy to get it to work. So I remember this movie as if it was yesterday, because at around 4 a.m. in the night, what I saw was these inflammatory cells coming in here around the region where we have all the dead cells. But look how they're also up here around the area where we have the live cells. So when we saw this infiltration of inflammatory cells into the tumor tissue, we are naturally wondering, what are they doing? Is that a good thing or is it a bad thing? So to address that, we need to take a step back and think about what are inflammatory cells really doing in the body. And this is an example where we infected a mouse with a fluorescently labeled red bacteria. You can see the bacteria is all over here. And when I start the movie, you can see that the inflammatory cells are going through the blood vessel and they're seeking out the bacteria. They're getting out of the vessel and they're running right towards them. And when you come here in higher magnification, what you'll see is that not only do they find the bacteria, in a moment one of these cells is going to come in, and you'll see how it engulfs the bacteria one by one, takes them up and clear them and get rid of them. So these cells are really very important for us in our defense against bacterial infections. See the cell come in here again. But they have additional function. In the case of wound healing, these inflammatory cells are coming in first and they're cleaning out the bacteria, but then the wound needs to close. And in order for that to happen, these inflammatory cells start a cascade at the wound healing process by sending out signals that make cells in the tissue proliferate, so they can regenerate the tissue that was injured. They make the blood vessel sprout, so there will be new vessels to this new tissue. And then they start stimulating the movement of cells to come together to close a wound. This is all something we need, but the tumor can hijack this process, and it does so very commonly, because these signals that can stimulate cell proliferation and vessel growth are the same signal that the tumor then can exploit to increase its growth, and the signals that increase movement and closure of the wound can be exploited to promote metastatic spread. So that leads us back to the chemotherapeutic study. What are these inflammatory cells doing after chemotherapy? So to address that, we started treating mice, normal mice and mice where we had genetically manipulated them so there was a reduced inflammation. And you can see when we treat with one of these drugs, the drug called doxorubicin, we can keep the tumor somewhat in check initially and then it really takes off. But when we remove the inflammatory cells from the mouse, we can keep the tumor in check for much longer. So we're not curing the cancer, but we're keeping it stable for a longer time. Another drug called cisplatin, again, it's not that effective in a normal mouse, but if you take away the inflammatory cells, now we start to see shrinkage of the tumor. So what we are learning from this is we can start to play around with different drug combination and reduction of inflammatory cells and get a better response to the drugs we already have available. The question is, and that leads us to what I really want you to take home from this is that there are many different ways that drug resistance can occur. The classical way of thinking of drug resistance is that there's mutation in a subpopulation of cancer cells, and that this mutation result in resistance to a drug. But there are additional mechanisms, as I showed you. Some cells may not even see enough drug, and there are now several labs working on trying to get drugs deeper into the tumor, into the regions of the tumor that didn't see it. And I also showed you that these inflammatory cells are secreting survival signals, generating survival signals for cancer cells. So that's another way we can try to think about how drug resistance and all these different processes are really coming together, and we need to address them together to cure breast cancer. Can we do even better? Here I've shown that we can inhibit the inflammatory cells. But I've talked about how they have several different functions. And these inflammatory cells that I talked about, the ones you saw in the movies, they're actually called monocytes. It's one specific type of inflammatory cells. You can think of it sort of as a teenager that grows up to become another type. The macrophage wants to get into the tissue. And in the tissue of the tumor, it gets these signals of wounds and becomes the tumor-promoting macrophages. But I also showed you that if there are signals of infections, it's a bacteria-killing macrophage. So what if we could make these bacteria-killing macrophages to tumor-killing macrophages? So a group of talented postdocs in my lab started thinking about this. It's a little bit like Clark Kent. He's ready to go. And he could be Superman, but the tumor takes him to the bar and it ends really poorly. Can we get him off those alcoholic tendencies and get him back on track to help us? And so what we did here was take macrophages and cancer cells, and they're exactly the same cancer cells shown here in red and macrophages in green, same cells in these two culture dishes. This one, we are leaving them to their own devices and let them have their normal stimuli, the normal signals that they're sending out. And as you can see, the cancer cells are growing happily. All these red cells are proliferating, and within 48 hours, they've taken over the entire culture dish. Over here, we are now giving the macrophages signals, so they think that they're a bacterial infection. And what you see is how they, one by one, attack the cancer cells. They kill them, and they eat them. And within 48 hours, we have almost completely cleaned up the petri dish of these cancer cells. So let me just show you here side by side. These are the same cells. All we did was trick them to think that there was a bacterial infection. So of course we want to try to see if we can take that into the patient. It's not trivial. There's a lot of side effects by triggering these bacterial infection signals. But I think it's worth a try because this is really dramatic, as you can tell. So I think that we are at a very exciting time. I've already shown you how looking at the tumor as a tissue and looking in real time is giving us insights on how we may be able to delay or even prevent relapse by getting the drugs we already have at our disposal to work better by attacking this immune cell infiltration that I've shown you. And there are several different labs working on this and several different approaches that are already in phase 1 trial because of the encouraging results from the mouse studies. But we want to go further, and we want to try to see what we can do to really address a metastatic disease and employ the ability of these immune cells that I've shown you, their ability to seek out and find the cancer cells and stop these signals of wound healing and instead activate the signals of cell killing so that we may be able to cure breast cancer. So I'd like to end by saying that I've never been more excited and inspired to be a breast cancer researcher than I am right now. I think we have some fantastic tools to our disposal. You may be aware that federal research funding is hard to come about at the moment. It has been, the research field has been pretty hard hit. And so therefore, I'm very grateful for all the local support I've received. We have a saying in Danish, and I'm from Denmark, that many small streams together make a big river. And I have received so much support here from the local community, from $50 donations and up to hundreds of thousands. And it's really been very enabling. That support had helped my research program come off the track, and together with the insights and help I've gotten from Janet Price and Joanne Marquette, really helped me secure the federal funding that David mentioned earlier so that we can take this research even further. And with that, I'd like to thank you for your attention, and I'll be happy to answer questions after the next talk by Nicholas Tonks.
Our next speaker is Dr. Nick Tonks. Nick is a professor at Cold Spring Harbor Laboratory. He's been at the laboratory for 24 years, and he has been elected a fellow of the Royal Society, and he's also a deputy director of our cancer center. Nick is a leader in the field of protein biochemistry, and his research is focused on a process called signal transduction, and he'll be talking to you tonight about this important process. He was the first scientist to isolate a very critical family of enzymes called protein tyrosine phosphatases, and these enzymes affect many different processes in the cell, and he's been harboring their biology to try to make an impact on breast cancer. Nick has developed a very innovative way to target a critical protein that Big Pharma has given up on in terms of developing therapeutics to impact breast cancer. And again, just as Mikala said, this is really a beautiful example of how basic research can step in and make a significant impact in helping to impact disease. So with that, I'd like to introduce Nick.
Okay, so thank you, David, and thank you all very much for coming. As David said, well, I want to, again, re-emphasize this point about the importance of basic research. And I want to talk to you tonight about the area of research that we work on, which, as David said, is called signal transduction. And that is the mechanism by which cells sense changes in their environment, such as growth factors, hormones, cytokines, etc., and translate those stimuli into changes in proliferation, differentiation, movement, metabolism, survival. So what I want to focus on is highlighting how breakthroughs in an academic setting, where we have been able to look at changes in these signal transduction responses that are associated with disease, and an understanding of the mechanisms of action of the proteins that underlie these changes in signaling, are leading to breakthroughs in development of new therapeutics. So I guess the overall focus of what I want to talk about tonight is, where are the next generation of drugs going to come from? So it all comes down to the issue of communication between cells. And some examples are shown on this slide here. And they include things like classic hormone responses, hormones that are produced in one part of the body and exert their effects on distant target cells, hormones such as adrenaline or insulin. Things like neurotransmitters that act in a more localized manner. Regulation of cell-cell contact, cells sensing their neighbors, and that leading to changes in proliferation and survival. And these are the sorts of processes that are disrupted in diseases such as cancer. So the cell is surrounded by a membrane that forms a barrier to the outside environment. But the cell doesn't ignore its environment. It wants to sense the changes, and it does so by the presence of these receptor proteins that can recognize the hormones and the growth factors and the other stimuli to which the cell has been exposed. And so this interaction between the stimulus and its receptor leads to changes in a series of proteins that cause changes in gene expression events in the nucleus that give you the functional response. And what I want to do is now show you a little video that was prepared by Cold Spring Harbor Lab's DNA Learning Center that illustrates a signal transduction pathway starting with a growth factor, platelet-derived growth factor, binding to its receptor on the surface of the membrane, and how that message is transmitted into the nucleus to give you the changes in gene expression that underlie proliferation. And interestingly, this is one of the signaling pathways that is disrupted in cancer. So let's hope this works.
A signaling pathway begins with the arrival of a chemical signal, such as a hormone or growth factor, at the cell surface. The gray structures sticking out of the cell membrane are receptors for these incoming signals. The signal, in this case a platelet-derived growth factor, here in purple and blue, encounters and binds to its matching receptor. A second receptor protein joins in, making the growth factor fit like a key in a lock. The binding of the growth factor causes the receptors to change shape. This change in the protein will be conducted through the membrane and into the cell's interior, the cytoplasm. The signal is conducted through the cell membrane into the cytoplasm. The binding of the growth factor outside the cell has caused the ends of the receptor, in gray, to intertwine and activate each other, shown as yellow flashes of light. Once active, the modified receptor ends interact with messenger proteins that will carry the signal through the cytoplasm. From our position in the cell's cytoplasm, we can see the ends of the receptor, in gray, being drawn together as the growth factor outside the cell binds. The receptor ends activate each other before binding an adapter molecule, shown in pink, and an exchange factor, shown in light purple. An important protein in this pathway, known as RAS, shown in red, then swings around to be activated. RAS, in turn, activates 3 white RAF proteins before another protein, shown in blue, moves in to deactivate it. RAS is a key switch in this pathway. Mutations in the RAS gene and protein are common in cancer cells. The activated protein, in pink, is transported into the nucleus through a pore in the nuclear membrane. The nucleus contains tightly wound coils of DNA, shown in green. The signal is passed to 2 other molecules, Phos and Jun, in yellow and pink, that team up to locate a specific gene along the DNA. Phos and Jun bind the DNA, starting the process of transcription. Other proteins are then called into play that unwind and open the DNA molecule so that RNA polymerase, shown in brown, can make a copy of the genetic information. The copy, called messenger RNA, here in light green, is packaged with a set of carrier proteins and leaves the nucleus. The cell will use this copy to make a new protein.
So that gives you an idea about a standard signal transduction pathway that starts with an interaction between a growth factor, for example, with its receptor, leading to the addition of phosphate groups to the receptor itself and other molecules that leads to an assembly of a multi-protein complex that can now trigger these cascades of phosphorylation reactions that lead to changes in gene expression and ultimately the physiological response. And then the class of enzymes we'll talk about in a second, the phosphatases take the phosphate groups back off again and can return the system to the ground state. But these normal pathways create signatures of phosphorylation events that underlie the normal state. And they are disrupted in cancer. And so you can end up with disruption of these signaling pathways, which causes disease signatures, which now allows you the potential for therapeutic intervention by targeting these changes that you find as a characteristic of the disease state. So one of the earliest examples in a cancer context, a very good example, is P210 BCR-ABL. And so what this is a chromosomal translocation where a protein tyrosine kinase, a kinase gene encoded on chromosome 9, is fused with a different gene on chromosome 22 to create this abnormal chromosome that is a characteristic of chronic myelogenous leukemia. It generates a protein that is not found under normal conditions, in which the kinase activity of c-Abl is aberrantly activated and underlies the disease. So here is an example where targeting this cancer-causing gene with a therapeutic could lead to a treatment. And actually, many of you will have heard of the small molecule inhibitor Gleevec, which is one of the first examples of a signaling, a regulator of signal transduction, a small molecule regulator of signal transduction that could function as a drug in a cancer context. And it had a very important impact on CML and other cancers. In the context of breast cancer, we've already heard about these growth factor receptor tyrosine kinases, molecules with the ability to transfer phosphate onto other groups that serve as the receptors for a number of different stimuli that the cell can sense. And they do so, as it said in the little video, by forming dimers amongst one another. For example, ERB2 binding to ERB1, ERB2 binding to ERB3. And this is the ERB2, the HER2 new molecule that is hugely overexpressed in certain forms of breast cancer, the HER2 positive forms of breast cancer. And the overexpression, the amplification of this underlies the disease. So this again is a target that is unique to the disease, to the cancer cell, that offers a mechanism for hitting specifically the cancer cell. And so we heard Laura talk at the very beginning about a different type of therapeutic intervention that involves antibodies, proteins that bind with very high affinity to specific molecular targets. And different antibodies have been generated towards the extracellular segment of the ERB2 HER2 molecule that prevent its signaling function and offer an approach to treating diseases in which HER2 is overexpressed. Actually, the most recent developments in this area have been very powerful, and they involve, there's multiple antibodies now that can affect the extracellular segment of these receptor tyrosine kinases. And actually, this has the advantage that you don't actually have to get the drug across the cell membrane inside the cell to have an effect. You're exerting an effect on the cancer cell from outside. But also by using this as a marker of the disease, some of the latest developments, such as this TDM1 molecule, involve putting a cytotoxic agent as a complex with the antibody and using the antibody to deliver that cytotoxic agent specifically to the cancer cell. And this is a new approach that is showing great success. Now, there are spectacular successes from these sort of approaches, but there are still challenges. One is their expense. These are very expensive therapies, the antibody-based therapies, but also the issue of resistance. Not all patients respond, and those that do become resistant to the therapy. So what we need are different classes of signaling molecules that we can target that will give us different classes of drug candidates that can be used on their own or in combination with these kinase inhibitors to try to overcome this resistance. And so the element of signal transduction that my lab works on is the complementary approach to the kinases, which are the enzymes called phosphatases. And so the signaling pathways are assembled depending upon the phosphorylation of proteins in those pathways. So kinases transfer phosphate onto the protein, and the phosphatases take the protein back off again. And the particular family of enzymes that I work on are called protein tyrosine phosphatases, and we're interested in trying to define their cellular function, establish links to disease, and really characterize these molecules as new therapeutic targets. The one I've been working on most is a molecule I purified first about 25 years ago while I was a postdoc at the University of Washington in Seattle, and it's a molecule called PTP1B, and it has two very interesting phenotypes. The first one is that when you knock out the PTP1B gene in mice, you get this amazing effect. This is a wild-type mouse fed a high-fat diet. This is a PTP1B knockout mouse fed the same diet. You can feed them the equivalent of hamburgers and French fries and they don't get fat. And when you're battling middle age like I am, this is a really interesting phenotype to be working on. So these mice are resistant to the development of obesity and type 2 diabetes that accompanies a high-fat diet. But also, this same phosphatase plays an important positive role in promoting signaling downstream of the HER2 protein tyrosine kinase that is the cause of HER2-positive breast cancer. So what this says is that we can look at these phosphatases as therapeutic targets for treating diabetes and obesity and treating HER2-positive breast cancer. So in the case of diabetes and obesity, where the kinase is responsible for switching the signaling on, and the disease is associated with not enough signaling coming from the insulin and the leptin receptors, inhibiting the phosphatase to block the back reaction will help to overcome the insulin-resistant state. In cancer, where you have a molecule such as the HER2 oncoprotein, where the action of the phosphatase is important for promoting the tumorigenic function of HER2, inhibiting that phosphatase will block that effect and may be an alternative approach to inhibiting HER2-positive breast cancer. So how do we go about looking at this? The standard way that people do this in industry is to generate small molecule inhibitors that target the active site of the enzyme, the point on the enzyme where it does its business. And so what they do is they will take proteins and treat them with huge libraries of chemical compounds and look at the ability of those libraries to inhibit the activity of the enzyme. They'll come up with candidates, and those candidates will be modified in subsequent rounds of chemistry that will give you a final molecule that will bind at the active site of the enzyme and inhibit its function. And many people tested that with PTP1B, and what they came up with, for those of you with a chemistry interest in the audience, were molecules that looked like this, that were mimics of their substrates. They came up with potent, reversible, selective inhibitors that were highly charged due to the presence of these groups here, which meant that they couldn't get across membranes very easily, which meant that they had very limited drug development potential, which meant that industry said that they were then undruggable. So what we've been able to do is to, in an academic setting, is to take that as a challenge. And really, because we work on these things to a much greater level, we can think deeply and differently about how to approach these problems. We've been taking different approaches to try to come up with inhibitors of this molecule. And we're making several different approaches, but I just want to briefly highlight one, because it's going to be a concept that you're going to encounter many times now in drug development efforts. This is what PTP1B, this is the crystal structure of PTP1B, what the protein looks like in molecular level. But most industrial efforts to generate small molecule inhibitors of the enzyme focused on a truncated version of the protein that represents this catalytic portion. But in reality, in the cell, it is present as a longer version that has a regulatory extension on it that targets it into intracellular membranes. So we asked the question, can we use this longer form here, which is more difficult and more technically challenging to make and to work with, but can we look and see if we can see things that will inhibit this form preferentially over that form, and look for things that industry could have missed? And we were able to do that. We came up with a natural product inhibitor called traduscamine, and it turns out it is an allosteric inhibitor of the enzyme. So what that means is it causes the protein to change shape. And the best way to think about this is that the phosphatase, PTP1B, is like a clamshell. In the inactive state, it is open. When a substrate binds, the phosphatase, the active site closes, and it gives you the active form of the enzyme. These allosteric inhibitors bind at a different site on the protein to the active site, but lock it in this open, inactive form. So it's a different mechanism that we now understand at a molecular level that allow us to inhibit this enzyme from a new approach. And from the breast cancer perspective, we've been able to test this in cell and animal models of HER2-positive breast cancer with some dramatic effects. And one of them is shown here. And this is looking at the ability of this compound, which is also called MSI 1436, to have an impact on metastasis to the lung in a mouse model of HER2-positive breast cancer. And so these are the control animals. These are sections through the lung of the control animals, and you can see the metastases that are growing in the lungs. But in the mice that are treated with the inhibitor, you can see that the metastasis is essentially abrogated completely. So through this approach, we have identified a new inhibitor of this phosphatase that is providing a new mechanism for inhibiting the activity of HER2 and inhibiting HER2's tumorigenic potential. And we are getting together now. About a week ago, we started a small company to raise the funds to start a clinical trial in collaboration with our colleagues at the Monterey Cancer Institute and also LIJ, with Dan Budman, who's sitting down towards the front here. He's one of the lead scientists over at Monterey Cancer Center. And we're taking this into the clinic right now. So this really is an exciting time to think that basic research is going to have a potential impact on the lives of real cancer patients. Going forward, we have been able to generate a new set of allosteric inhibitors that are structurally related to but distinct from this traduscamine molecule, that show better oral bioavailability. They're more potent inhibitors. They're a set of molecules that we can now take forward and optimize as the next generation of allosteric inhibitors of PTP1B, and they are following on behind for the next phase of clinical trials. So in terms of take-home messages, I would just like to emphasize, I think, again, basic research has a crucial role to play. We're just not smart enough to know where the next great breakthroughs are coming from. And it's the basic research that unexpectedly gives these breakthroughs. As Mikala pointed out, the fluorescent proteins that are such huge importance in her research in looking at cell-cell interactions in live animals. The current economic climate indicates that industry can't do the kind of basic research that we can do. So there is an opportunity here to do things differently to industry, but then to synergize with industry in taking new candidates forward for drug development. I hope you'll think about the signal transduction pathways as fertile ground for the identification of new targets in cancer and other diseases. And what we've been able to show specifically in the research that we're doing is that just because industry thinks that a particular class of enzymes are undruggable, if you know the function and the mechanism of action of these proteins in detail, there's the possibility to generate novel inhibitors that allow you to manipulate these highly validated targets and thereby open up new avenues for treating major diseases. And so, again, what we're hoping is that this will offer new approaches for treatment of not only of breast cancer, but also diabetes and obesity, and in the cancer context, specifically perhaps addressing issues such as the resistance that develops to the existing therapies. Finally, one of the nice things about working at Cold Spring Harbor is, as a principal investigator for a long time, you get to work with some really talented young scientists. The person in my lab who's been doing all of the work on this new approach to inhibiting PTP1B is shown here. It's Navakrishnan. He's an extremely talented postdoc with a very bright future ahead of him. These are collaborators that we've worked with on the characterization. This is the small company that we've started to try to take this inhibitor into the clinic in collaboration with Dan and Myra and the Monterey Cancer Center. And also just remind you of the DNA Learning Center here, and there's a very interesting website. There's lots of information that you can get on various aspects of biology, various aspects of gene regulation. And finally, I would also like to mirror the thanks for the support of the local community for the research that we do. These really are very difficult times to carry out research, basic research, but also it's hard to do this kind of research that bridges the gap between the academic setting and industry. Neither side really sees it as its responsibility to fund that kind of research, and so the support of the local community has been hugely important to helping us to make the progress that we've made, and I'm very grateful for your support. Thank you all very much.